Welcome to this explainer. Today, we're unpacking something absolutely massive in the world of clinical genomics. If you've been following this space at all, you know that time is the ultimate bottleneck. So we're going to break down exactly how scientists engineered a world record-breaking pipeline, synergizing hardware, software, and wet lab biology to deliver life-saving answers in a matter of hours instead of weeks. Picture this. You have a critically ill patient, maybe a newborn in the NICU, desperately needing answers to a biological mystery. Now imagine getting a Guinness World Record-setting full human genome sequenced in just 5 hours and 2 minutes. This isn't just theoretical anymore. This exact milestone was actually achieved by a team of researchers who completely re-engineered the diagnostic pipeline from the ground up. To understand exactly how they pulled this off, we'll map out their structural blueprint. We're going to cover 1, the race against time, 2, breaking the compute bottleneck, 3, the AI and cloud stack, 4, optimizing the wet lab, and 5, filtering for actionable insights. Let's get right into it. Section 1. The race against time. In critical care environments, every single minute counts. Diagnostic speed is basically your primary lever for clinical intervention, but standard genomics has historically suffered from some pretty dangerous latency. Just think about this incredible paradigm shift. Historically, a quote-unquote rapid sequence took a painful 56 hours. Over time, teams optimized that down to 48, then about 14.5 hours. But when you're dealing with acute heart failure or unexplained neonatal seizures, even a 14-hour window is a massive critical delay. This new approach crunched the entire end-to-end diagnostic time down to a staggering 7 hours and 18 minutes for a specific clinical case. So how did they do it? Let's move to Section 2, breaking the compute bottleneck. To compress the timeline this radically, the team had to solve a really interesting problem. Here's the wild part. Running a high-throughput platform like the Prometheon 48 actually generates data way too fast for local computers to handle. We're talking about a massive data deluge of up to 1.8 gigabases per minute. That instantly chokes a standard local compute tower. The sequencing hardware physically creates raw signal files faster than local machines can process them, which used to result in hours of wasted overhead time. Their solution? A really clever hardware-software handshake. By funneling all this data straight to Google Cloud, they bypassed the local bottleneck entirely. They used a fast file compression standard called VBZ and uploaded the raw signal files in batches every 3 minutes. This prevents network traffic jams and keeps the cloud compute instances constantly saturated with data. It's massive parallelization for near real-time processing. That brings us to Section 3, the AI and cloud stack. Stripping away those latent minutes relies heavily on an integrated tech stack powered by extreme GPU acceleration. It's a brilliant setup. They deployed an absolute army of cloud hardware. You've got 16 instances packed with NVIDIA V100 GPUs for real-time base calling using a tool called Guppy, running right alongside Minimap 2 for alignment on parallel CPUs. From there, the data flows directly into 14 cloud instances loaded with NVIDIA P100 GPUs for small variant calling. And let's focus on that variant caller for a second because it's a total game changer. The AI tool called Deep Variant shifts away from traditional Bayesian statistical models. Instead, it uses a convolutional neural network. It literally visualizes genomic features, things like base quality and strand orientation, and encodes them into multi-channel pile-up images over 221 base pair regions. So, by analyzing these high-dimensional images, this deep learning network spots mutations basically the same way your phone spots a face in a photo. This allows the AI to accurately identify mutations in super tricky, low-complexity regions, like the homopolymer errors you often see in nanopore sequencing, without needing time-consuming manual hard filtering. Moving right along to Section 4, optimizing the wet lab, because honestly, it wasn't just the computational software that got an overhaul. Biology literally had to keep pace with silicon. Think about it. A standard protocol just wasn't going to work. The team optimized the sample prep to extract high molecular mass DNA in just 50 minutes. Then, instead of prepping 48 separate sequencing libraries sequentially, they scaled up to an 8-way parallel reaction with higher DNA input. This gave them the exact yield they needed to load all 48 flow cells simultaneously. Now, 37. That number is huge. By skipping the ligation and cleanup steps normally associated with sample barcoding, they directly shaved 37 crucial minutes off the clock.
And if you're wondering whether mixing non-barcoded samples ruins the data on reused flow cells, they tested it. Even with a simulated 1% physical carryover, variant calling maintained peak accuracy. They gained vital speed without sacrificing an ounce of clinical integrity. Finally, we're at Section 5, filtering for actionable insights. Because at the end of the day, you haven't really saved time if the physician is left drowning in a sea of data. Standard pipelines often return well over 100 potential variants, which is practically a recipe for physician burnout. But in an acute setting, clinicians need clearly pathogenic, actionable variants in established disease genes. So they engineered an ultra-rapid variant filtration schema. It's a score-based system that evaluates criteria like biallelic inheritance and predicted deleterious impact, requiring a weighted threshold of 4 or higher. The results are pretty amazing. A standard pipeline usually surfaces around 147 candidate variants for review. This AI-driven system drops that number down to just 31 highly actionable variants. That's a massive 79% reduction in volume. It completely mitigates burnout and allows doctors to hone in on the exact genetic culprit almost immediately. Which brings us to this profound realization from Dr. Ewan Astley. The whole goal is getting an answer before the end of a hospital ward round. This brilliant integration of wet lab efficiency and AI-powered cloud computing literally transforms the diagnostic odyssey from a harrowing journey of weeks into a single hospital shift. So as we wrap up this explainer, I want you to think about the true impact of this ticking clock breakthrough. As AI continues to compress the time to definitive therapy, how will near real-time genomic insights fundamentally reshape the future of acute medical care? What becomes possible when we can solve life or death biological mysteries before the sun goes down? Thanks so much for joining me today, and I hope this gave you a powerful new perspective on the future of human health.